So we're back to our usual wet lab format. So you get the handout, and we'll go through the same sequence of material. For today's wet lab, we're going to discuss some of the methods that used for detecting the micronuclei of radiated samples. And we'll go through a little bit about background of this method. If you decide to do it yourself, what kind of equipment you need, the supplies. And we have a particular procedure that's related to the flow cytometry method. And then at the end, we're going to divide the group into two because we also like to show you some of the traditional way of measuring the micronuclei. That's by the microscopic method. And so because of that, we're going to have two places, one upstairs in the flow cytometry lab, the other one is down in the basement where you did the chromosome spreading before. So that my lecture is going to be a little bit short today, so that will allow us enough time to do this walk back and forth between two places. Now, for background, we're going to discuss two different types of micronuclear assay. Like I mentioned, one by the microscopic method, the other one's by flow cytometry. Also, a little bit about how this method can be applied for different kind of applications. So the first thing is that what is micronuclei? If you remember that several months ago we did the chromosome spreading, we all look under the microscope, we try to score the dicentric chromosome aberrations. Okay, so essentially micronuclei is a leftover of the other part of the dicentric, and so you can consider the micronuclei as a part of the chromosome aberration method. In this case, we know there's many different ways that you can get the chromosome aberration or the dicentric chromosome. In this case, we say there's two pieces of chromosomes that they are pre-replications, a G1 phase of cell cycle. They're hit by radiation, in this case, one break on each piece of chromosome. And then when they try to recover, they go through this illegitimate reunion. Then you have that piece of chromosome stick to the other one, so you get a two centromere. And during the cell division, you get the dicentric. Then the other piece left over, there's no centromere. They call it acentric fragmentation. Okay, and but this piece of chromosome can be vary in different lengths. Okay, it could be as long as a whole piece of chromosome, as short as a very small dot. So when you look the dicentric and this fragmentation last time, you saw them in a spread form, and you actually saw the actual chromosome. But there's other ways you don't have to go through that chromosome spreading. You can look at the cells and to find out that particular fragmentation. That's one method you can do using one of the micronuclei assay analysis. And this is gaining more popularity in industry as a way to measure the mutagenesis, carcinogenesis, or genotoxicity of many different types of chemicals. So it has been applied to detect both the clusterogenic and the angiogenic effect of testing agent. This could be anything, because for instance, if you have a new compound or you want to see seek any kind of FDA approval, probably you have to do this type of analysis to prove that agent is not toxic or cause any kind of mutation or carcinogen. In case you're not familiar with these two terms, if you're not in the toxicology program, the clusterogenic is any substance or process causing chromosome breaks. Certainly, radiation is one of the agents that can cause this clusterogenic effect. Angiogenic are sort of slightly different. Those are the agents which affect either cell division or the mitotic spindle fibers, result in the loss or gain of whole chromosome. So the difference between these two are the one's whole chromosome, other is a part of a chromosome, and thereby induce an aneuploidy. That's why the angiogenic term is coming from. Now, many agents, including radiation, they can be both of them. For example, X-ray is very good of causing chromosome breaks, but it can also induce this type of thing. Vice versa, many chemicals are involved in the spindle fiber effects. For instance, chemotherapeutic drugs such as vinblastine, vincristine, or taxols, they all have effect on spindle fibers. They will show more of an angiogenic effect than clusterogenic. But on the hand, for instance, cyclophosphamide, which is mostly angiogenic, mostly carcinogenic, not angiogenic. So almost any type of chemicals can be involved in one of these or both of the effect. Now, it's gradually becoming a trend that the micronucleic acid will replace the chromosome spreading, what we call the metaphase analysis, as a screening choice. And basically because it's more cost effective than the conventional chromosome aberration assays. Okay, we'll tell you now why it's cost effective a little bit later on. Now, the method that we're going to describe are involving the reticulocyte or red cells, and that's an in vivo micronuclear assay. And this is a very, very important development. It's mostly made by the Lightron company just outside our campus. And this is usually conducted in mice because many of the toxicity tests will involve a final stage of any kind of animal. Mice is one of the most popular species. It's especially important because there's no in vitro alternative that can be validated in this moment. So that because this method would measure the micronuclear production directly out from animals, so that effect is the truly in vivo effect. And so that's how it's gained more and more popularity. And the cells that involve this type of assay are usually either from the erythrocyte population, from either the peripheral blood or from bone marrow. Of course, the other type of cells can also be used for micronucleic acid, but not necessarily by flow cytometry method. With a microscopic method, you can apply almost any type of cells for the micronuclei formation. Now, we said earlier there's two types of method that you can apply. One is called a cytokinesis block micronuclei method, or the CBMN, and most time you will use the lymphocyte from the peripheral blood or any kind of cell line or fibroblasts, even tumor cell line, and to measure the degree of micronuclear formation. The second one involves the flow cytometry. In this case, it's only detect the micronuclei from red blood cells. Now, we said that the micronuclei is relatively simple and cost-effective, and for the microscopic method, everything can be explained, all the procedures and principles can be explained by the simple slide here. In this case, the critical chemical that used is cytokinin B. It's an actin inhibitor. What it does is that it inhibits the cell division by blocking the cells between metaphase and anaphase. That's where the two nuclear materials start to separate into two cells, but without the action of spindle fibers, and the two nuclei would stay at that. It's sort of frozen at that stage, so you won't be able to get two separate cells. So the whole process involved in most cases, this is the use of in the lymphocyte. First involved mitogen stimulation, so that you induce the proliferation of the lymphocyte. And that usually will carry on for almost about 44 hours. So if you look at the time scale, it's divided into day 1, day 2, and day 3, 24, 48, and 72 hours, because normally we assume that the cell division time is about 24 hours. Every 24 hours, cell will divide once. So you allow the cell to divide for lymphocyte, but even with the best mitogen, you're probably not going to get all the lymphocyte to divide. Usually you can get about 50% or even more than that. You allow the cell to go through its first division, but before they reach the second division, 48 hours, you apply cytokinin B. So this particular method that applied about 44 hours, you start the cells from the second division. So then you allow the cell to go on for another sort of cycle, and you get this kind of binucleated cells because they couldn't go through. Or you may still have cells with single nuclei or mononucleated, because these are the cells who never even went through the first division. And then so you have the first division, second division. And there, if you have a micronuclei, you can see a small dot of a nuclear material stand by, say, Ginza stem. And the reason that you have this kind of thing is that it could be that you have some inherited micronuclei formation or background amount of micronuclei. So this could be serve as a sort of background measurement of your population. So this is just a picture of what you can see under the microscope. In this case, the cells are blocked at the metaphase, but there's no micronuclei. You have additional nuclear material as a micronuclei. Now, we talk about the two different types of micronuclei. One is the antigen, the other one is clastogen. How are you able to separate these two types? You can take the combination of this particular method using cytokeratin B and the FISH techniques. For example, you start with a lymphocyte and you stimulate them just like the previous slide shown. And if you go through the regular process, you have giemsa stain and you plus the cytokeratin B, you can find the micronuclei. But you don't know the size of this micronuclei. This could be a tiny amount of DNA fragment. This could be a whole chromosome. How are you able to tell that's a whole chromosome, which will belong to the antigen category if the treatment is by this type of chemicals, or this could be clastogen, small amount of nuclear material. So you can introduce the FISH technique, the fluorescence in situ hybridization. In this case, use a probe. The probe belongs to the centromere. I don't know if you can see those green dots inside the nuclear region. Those are the actual centromere. So if you have 20 pairs of chromosomes, theoretically you're going to have 20 dots there. Okay. Now, if you compare these two types of micronuclei, one with the probe, the other one without the probe. The one without probe means what? It's an intact or part of a chromosome. It's only part of a chromosome. Otherwise, you should have the probe there if it's an intact chromosome. So the definition of these two types of chemicals is one, you induce micronuclei contains whole chromosome. So if you see the probe shining up on your micronuclei, then you know that the chemical will cause this particular type of response as an antigen. If there's no probe on the micronuclei, then you have the clastogen effect. So this is where you can separate these two, and this is actually the way that been applied in industry, many places to define the type of chemicals that are in this category. So when you do this kind of microscopic measurement, there's certain criteria. It's not very hard to follow this criteria. What is micronuclei? What is not micronuclei? First, the diameter of micronuclei should be less than 1/3 of that main nucleus. So you cannot have the same size of a nuclear region called micronuclei, because when you do the cytokeratin B treatment, you don't necessarily just get the 2 nuclei. Sometimes you get 5 nuclei, 6 nuclei, 7 nuclei. So how do you know those are nuclei, not micronuclei? Follow this definition. They have to be about at least it has to be less than 1/3 diameter. Secondly, micronuclei should have similar staining intensity as the main nucleus. And you say, what the problem is that? The problem is that if the cell also has gone through apoptosis, and you all know that the apoptosis, you get the fragment of the nuclear material, and most time you can see this thing called the apoptotic body. It's also a very dense stained spot in the cytoplasmic region. And in that case, those are not micronuclei, those are apoptotic body. So they went through a completely different kind of process and mechanism. So for micronuclei, they should have similar staining intensity. And finally, the micronuclei should be separate or very marginal overlap with the main nucleus. And that's, again, if you have a multinuclei but not micronuclei, this is a way to separate them. And sometimes there's additional criteria, but it's not applicable to every type of cells and conditions. That is the relationship of the 2 nuclei. And sometimes it's defined as that you have to have some kind of contact of these two. But if you remember the slide I showed you before, they are separated. The idea was that because the action of cytokeratin B is to inhibit the acting reaction to prevent the pulling of the thing apart, so that you must have some kind of connection filament there. But it's not used, I think, universally for defined micronuclei. So anyway, you can use it or you don't use it. In this case, certainly you can use that criteria, and this is truly main nuclei, that's micronuclei. So we said that this micronuclei, microscopic micronuclei method is gradually replacing the chromosome spreading for some of the reasons here that it's a very rapid method. In 3 days, you get a result. And for those of you who have done some of the chromosome spreading may argue with me, well, we can do the same thing in 3 days, so what's good about it? But the problem is that with micronuclei, you can get a final result in 3 days. With chromosome spreading, usually you take another 4 weeks to finish the scoring. But micronuclei, you can score them right away. And it's relatively cheap. You really don't need much of the material at all. Tissue culture medium, cytokeratin B, they are relatively inexpensive. And the other part is the manpower is much cheaper for micronuclei measurement than the chromosome spreading. Simplicity, almost everybody can be trained. It doesn't require any special training to do this kind of measurement. Most importantly, the statistical power. You can score hundreds of micronuclei versus most of the cases you can only get few chromosome spreading, and the result from the few chromosome spreading sometimes is not statistically significant to tell the differences, especially you are looking for minor changes. But with micronuclei, you are able to really increase the sensitivity of the detection method. However, there's some disadvantage of using micronuclei. First, the very obvious thing is that not all type of chromosome aberrations are detected because you only look for a very particular type of chromosome aberration. That's acentric fragmentation. Even you combine that with a fish, you can only get the other type of measurement. And the most troublesome part of this method is that you have to use cytokeratin B, and sometimes they are toxic to the cells. So the cytokeratin B itself can induce certain amount of micronuclei formation. So it depends on the additional treatment, other type of treatment. You might have a very high background, which you're not able to detect the differences. For example, when you go to the lab, you find out that you have to use different amount of cytokeratin B to get the micronuclei. Lymphocyte usually is more resistant to cytokeratin B than some of the tumor cell lines, so that you really don't have a uniform method you can apply to, say, even minimize the toxicity from cytokeratin B. We said that this method is become more and more popular. This just lists some of the examples of how micronuclei assays used for a variety of studies. And most of these are made by the microscopic method. First, it's most important to this U19 study is the biological dosimetry, because it can measure very low dose exposure, and especially with the general population without carrying the film badges like most people in this room does. And this is a great way to estimate the radiation exposure. Also, because of sensitivity of this method, that one can sort of pre-screen some of the cancer, potential cancer patient. For example, the lymphocyte isolated from smokers and showed a higher level of micronuclei, and the same population, when they looked upon at a later stage, found that they have a higher incidence of lung cancer. So this could be used as one of the indicators for the potential cancer incident. Similarly, that the arsenic exposure will increase the mucosa, the oral mucosa, and the bladder cancer. That again can be picked up at a much earlier time by the micronuclei incident. Other things, for instance, the pesticide effect, that sometimes very difficult to get the end result determined, but using micronuclei, one would see a higher level found in the children of those exposed to a pesticide. And this is purely academic research of whether the reactive oxygen species can also increase that. There certainly is one of the cluster gene type of reaction. And finally, for the radiation therapy, because it's individual variations among the patient in terms of their response to therapy. This could be another way of measuring their individual sensitivity by taking the blood sample, look at the micronuclei, and found that there's some correlation between the inherited radiation sensitivity and their response to the therapy. This is an ongoing study, so I really couldn't say that this is a sort of a method you can apply routinely for all patients yet. Now, a few words about the comparison between micronuclei and the chromosome spreading in terms of dose response curve. We have discussed about the dose response curve for instance, the dicentric. You have a very, very defined linear quadratic relationship. Once the dose increase, you get a dose square response. You can get this upward turning curve. But for micronuclei, we still don't know exactly what kind of dose response curve we will get. For example, whether it's linear, with a curve. Actually, at this moment, I think some of the study from Dr. Yuqiao Chen's lab showed that depending on which population cells you look at, if you look the lymphocyte, it'll give you this type of response. But if you look the red blood cells, it seems to give you the linear response, and they still don't know exactly why there's such a discrepancy between the two. And secondly, that for chromosome aberration, you can measure this kind of damage at the much longer after the exposure, several years up to. But for micronuclei, most of this measurement need to be done within week period. So you cannot take a sample from the early exposed population. And also the timing of sampling is very critical. If you don't take the correct timing, you're not able to get that maximum sensitivity. So much for the microscopic measurement. Let's talk about a little more about using the flow cytometer to measure the micronuclei. There's some advantage of using flow cytometry. You can do even much faster scoring. You can do these things within few hours after sampling, if you could, versus you still have to wait about 3 days. You can measure several million, 10 million, whatever number of cells you like to get. So that will really increase the sensitivity, so that reflect to a sensitivity of measuring a very small changes in micronuclei frequency. You can get down to 0.1, 0.2% of a micronuclei versus a microscopic measurement. You have to have at least a few percentage changes in order to qualify them as a significant difference. But the disadvantage is that this can only be done through the reticulocyte from the peripheral blood. And the criteria for such measurement is depend on DNA content. Looks very simple, but in practice, you are facing several problems. For example, if you do a DNA staining of your red blood cells and put on the flow cytometer, you get a display of DNA content measured by propylene iodide and the cell count. You get this some kind of distribution. So question is that, where are the micronuclear cells? So where's your micronuclei? Okay. The problem is that you cannot just simply determine the micronuclei by their DNA content, because most of peripheral blood is made of many different types of cells. So you have to make sure that you are looking at the correct type of cell subpopulations. In this case, if you look at this whole blood are simply divided into white blood cells and red blood cells. Certainly you don't want to look at anything from white blood cells. There are lymphocyte, monocyte, granulocyte, those kind of cells. But even within the red blood cells, you would have, say, populations of immature red blood cells which still contain DNA, because the mature blood cells doesn't have any DNA or doesn't have a nucleus in it. So you don't want to look the cell population from this group. Also, you have platelet. They may also contain some DNA, and that will give you a false reading. The only part of the cells you want to look at is reticulocyte, mature reticulocyte, and look at the amount of micronuclei. That is, these mature red cells, they don't have any nuclear material in it, but anything that contains DNA material, that's probably the representation of a micronuclei. So the Lightron laboratory, and it's actually sort of supervised by Steve Derdinger, spend most time to develop this method. So what I'm going to do the next about 5 or 10 minutes is go step by step to explain how this works. First, you want to eliminate those cells you don't want to look. So the first selection is to only look the red blood cells. And the good thing about red and white cells is that you can simply use a physical parameter to separate them. So if we look at the forward scatter distribution, basically the representation of the cell size, and versus side scatter, it's a representation of the cell surface structure or granularity of the cells. And you can define a group of cells, they are actually red blood cells. If you have done this kind of blood analysis by flow cytometry, then you know your lymphocyte, your other cells will be way above here. So by using this method, you can isolate just the red blood cells. But within the red blood cells, you still have the mixture of these cells. So how are we going to eliminate those cells that you don't want? So in this case, you rely on a marker, the marker that can recognize the platelet, the marker that can recognize reticulocyte. In this case, the marker that can recognize the platelet is CD61. The marker that can recognize the reticulocyte is CD71. And plus, you can look at the DNA content of the immature red cells. The red cells can still contain nuclear material. They have much higher DNA content. So by looking the display, and by the way, this is set up by Stephen Derdinger. Of course, if you are very familiar with flow cytometry, you can have your own protocol set up. But since this is sort of included in their kits, they'll sort of suggest protocol. So I'll go step by step and use his protocol. So in case you want to do it yourself, you buy the kits, then it's very easy for you to follow. And they're the logic of thinking. So the first thing is that you want to eliminate these cells. So you look at the propylating iodide DNA content versus the CD71, that's the reticulocyte. And since the immature cells contain much higher amount of DNA, so it will show the DNA content in this area. So you've put an area, we call a region, say I only the first step is that I only want to look at the red cells. The second step is that I want to look at the red cells, but not those immature red cells. So I will exclude those, I will include these things. So it will be single cells, total mature red blood cells. Okay? But they still include these two types of cells. So now what you can do is that you can use a marker to define whether they contain the platelet information or whether they can contain the reticulocyte information. And this would be the cells that have the expression of the platelet. So they are the platelet cells. Okay? So we don't want them. So we first say we want these cells, not those. So this is region. And secondly, say we want everything else but not this group of cells. And finally, we look at this particular display. This display looks very similar as that. With the exception, we only display this group of cells here. We already exclude that population. And then we say we only want to look at the reticulocyte. So they have the CD71 positive cells. So it's finally this group of cells which we eliminate the white cells, we eliminate immature red blood cells, we eliminate the platelet. Only these cells will look at their DNA content again, and you get this kind of display, which you're going to see now in the lab demonstration. That is, you display the DNA content versus the expression of the reticulocyte. So if you look at this large red box, they are all reticulocyte because they are all expressing this reticulocyte marker. Then if you look at the x-axis, it's a DNA content. These are the cells reticulocyte without DNA content because the mature red cells, normal mature red cells, they don't have any DNA. They are here. And here are the cells reticulocyte with small amount of DNA. So that's your micronuclei. Okay? And Then if you look at the numbers here, these numbers can be divided into 4 quadrants: upper left, upper right. Those are the reticulocyte. So the total number of reticulocyte will be that number plus that number. And then the micronuclear reticulocyte will be 571. So if you divide the 571 by the sum of these two, that will be the percentage of micronuclei you have. For this particular sample, this the mouse blood radiated, actually the animal was radiated for 1 gray gamma ray, and you get the percentage of reticulocyte about 2.5%. Okay, and later on I'm going to show you the one that without any radiation. Then you can see a significant difference between the radiated and non-radiated percentage of micronuclei. So this is essentially the method of measuring the micronuclei from red blood cells. So if you decide to get into this kind of research, like we always list what kind of equipment and the reagent do you need. Equipment, first you have to have a good tissue culture laboratory. That is primarily involved for the microscopic micronuclei measurement, or the CB, the cytokinesis blockaging. So you need a sterile culture hood, incubator, a microscope, pipette, those kind of things. Certainly you need a radiation source. Then if you want to do the other type of micronuclei measurement, you need a flow cytometer. In terms of supply, those listed here, the 5 of them, are for the microscopic measurement of micronuclei. The last one is for the flow cytometry measurement of micronuclei. You have to purchase this staining kit from Litron Laboratory. They have the intellectual, I think, a property patent for it. I'll tell you specific things about their kits that you're not supposed to duplicate yourself because they have the patent. With that, and the instruction pretty much follow those few slides I gave you a few minutes ago, you should be able to measure the micronuclei. Now, this part of the procedure I'm not going to describe you anymore because you're going to see them in the basement lab, and Irina Novak is going to give you some demonstration, also show you the final product of the micronuclei measured from human lymphocyte. So the first few steps, I'm going to show you how you're going to go about the flow cytometry measurement of micronuclei. Now, you purchase the kits. With the kits, and the kit will contain several ingredients. They include the red blood cells, the mouse blood cells, and the DNA staining, in this case propylene iodide, the marker CD71 conjugated with 50, and the marker for platelet conjugated with PE. Okay, then you just have to prepare and get these 3 essential calibration samples to start with. So the first sample would be the red blood. You add the DNA dye, but without the reticulocyte marker, but with the platelet marker. Second sample, you have to have the parasite infected mouse blood, and that's the critical ingredient for this kit. Parasite in this case is malaria. Malaria infected blood cells will contain the DNA from malaria, so that you have large population red cells contains very small amount of DNA. It's a detectable small amount of DNA, and that will allow you to use that as an internal standard, which is different from your normal immature red blood cells, but very close to the micronuclei DNA content. So in this case, you have the DNA PI, you put in the CD71, 50, but nothing else. The primary purpose, if you do flow cytometry, is to do this color compensation. So make sure that you don't have overlapping of the 2 markers. And third one will be, again, with parasite infected mouse blood cells. You have the DNA, you have both markers to allow you for a final setting of the instrument. So how do you go about it? Of the 3 separate calibration sample. I'll show you this thing again to sort of refresh your memory. Remember the stepwise elimination. First, we get rid of the white cells. We get just the red cells, but we only want to look at the mature red blood cells. And then we exclude the the platelet, and finally only look for the reticulocyte. But with the same principle, if you look at the red blood cells with DNA content without CD71 but with the platelet, what you see is that yes, you still have this high DNA content cell, you eliminate them. But on the other hand, you don't have much of the CD71, which is a reticulocyte marker, and you see a few spots here. This could be just the contamination of your sample, or could be the electronic contamination from your instrument. Nonetheless, this will be your sort of background count. Usually it's very, very low, no, less than 0.01% of the total event. But you do have the marker here for the platelet, so that will allow you to put the platelet into a proper position so they don't influence your measurement of the reticulocyte. And you do a final gating of just the reticulocyte with the reticulocyte marker, which we don't have anything, but without the platelet. So the final product of that red blood cell, you see that everything is sort of housing this negative, double negative area. You don't have much of the DNA content at all because you already eliminated them, and you're not supposed to have any micronuclei. And you get few spots. Like I said, this could be your the total background. Now your second sample will contain the parasite-infected mouse blood cells, which you have a DNA, you have the reticulocyte positive marker, and then you get this kind of picture. First, you have those red blood cells, immature red blood cells with normal DNA content, we exclude them. But since you put in the malaria-infected red blood cells, so these are the DNA from the malaria, the parasite-infected cells. And the ratio between the parasite DNA and the normal or immature red cells DNA, the ratio should be somewhere around 1 to 300. So actually it's in the neighborhood about 200 or 300 to 1. And remember the definition for the micronuclei, when you look at the microscope, it has to be about less than 1/3 of the diameter of your nuclei. So that when you do this kind of gating, you can also use that criteria so that your DNA content will be in that approximate neighborhood. But anyway, we just follow it. So you have this additional information here. You exclude the immature red cells. You don't have the platelet marker, so you don't have anything here, but you do have reticulocyte plus some of the parasite-infected red blood cells. You look at the final product, you see micronuclei, you see the reticulocyte without micronuclei. Where do you get this micronuclei? From the parasite, okay? But this is sort of like your internal control, indicating that you are able to measure the small changes in DNA content from the control. There's a good separation between your no DNA content at all to some of the minuscule amount of DNA in the red cells. And even though that's not induced by any kind of treatment, but this is from the malaria-infected DNA. Then the third sample, you have the parasite-infected mouse blood cells. Put in the CD71 marker and put the antiplatelet marker. What you see is again, you have the DNA here, we exclude them. But when you look at the CD61 and the CD71, now you see the platelet. You want to exclude that one and get that area and look your final product. That's your micronuclei. Now I know you didn't count, I didn't count either, but if you really count, this area will contain less dot than the one I show you. Why? Because you eliminate platelet. So platelet, they do give you some background count. So that's why you have to include them. So it's actually less dot here than the one without the platelet marker. Now finally, you take the real sample, you take the blood from unirradiated mouse, and you go through the same process. You have the DNA content, CD71, CD61. what you get a few spots here. You have majority of the cells in this area. These are reticulocyte by definition. And you get a 15 count here. You get about close to 6,000 count there. Then if you do a calculation, this will be your background micronuclei count, and the number is about 0.02 or 0.18. So that you have a range of 10 to 100-fold difference between no radiation and the 1 grade irradiated mouse blood, so that you really have a good range of measuring the amount of micronuclei. And in this case, you can also see that you can increase the total count. For instance, we didn't collect a large amount of events. If we collect up to say 3 or 4 million events, then you will increase this substantially. So this is basically how it measures. It sounds like complicated, but fortunately we have the program every set up. All you have to do is just put on a sample and you get your result. Now, how about human blood? You say, well, everything's fine with the mouse, but if you get into this nuclear terrorism, now you worry about the human being. How do we use this method applied to human blood? Basically, it's exactly the same principle. At this moment, the ideal kind of control would be parasite-infected human blood cells, which this company Lytron hasn't made it yet, actually. But if there's a commercial value here, you should be able to get those kind of blood relatively easy. Just make a trip to the Southwest Asia, go to the jungle area, and don't laugh at me, because actually malaria was a very common disease, because of my father's generation. Most of my uncles, my father, they all got malaria, because if you were in the army during World War II and fought enemy in that area, everybody got it. It's not a pleasant disease, but it's not totally lethal. So you can get blood from there if there's a commercial value. But unfortunately, we don't have that. So we have to just get by with the mouse blood, the parasite infected mouse blood again. But since we are doing this kind of thing for a different purpose, we have to use a different instrument, because we also like to sort this micronuclei out, so that we're able to study exactly what the component of DNA or chromosome in those micronucleated cells. So also you have to know that if you have different kind of instrumentation, you have always run this kind of a control. So in here, again, you look for the single cells, and if you only look at the DNA content, you have several peaks. That's your immature human red blood cells with large amount of DNA content. This is from, I'm sorry, this is not human, this is a mouse again, but we use as a calibration for the instrument. This is DNA content of a malaria parasite. And if you look for the expression of reticulocyte, you have a large amount of reticulocyte. Also, you have the platelet, and we follow the same principle, that we only look for this group of cells, and we only look for cells with response with positive reticulocyte marker. And finally, this is a group of cells we want to look for the micronuclei, and this just shows you with the malaria infected DNA that you're able to measure this type of cells. So if we put a human blood, this is drawn from a patient that received the radiation therapy, because you cannot radiate human for your purpose. So, but we are able to get the blood from patient, and we put through the instrument. Indeed, we are able to not only detect the small amount of micronuclei. In general, it's less than what we got from mouse. But we couldn't say the absolute production is less than mouse, because the dosimetry is very difficult to calculate in this case, because a patient didn't receive a total body. This is local radiation. But on the other hand, we were able to isolate these cells for other studies. So that's as much as I want to talk about in terms of the lecture. Then, like I said, we're going to divide the class in two groups.